Hello, and thank you for tuning into this video. Today we're going to be walking through the genomics variant analysis workflow on the FormBio platform. So for the purposes of this demo, we're going to be conducting a scientific research question, hoping to find variable positions in our human patient data relative to a human reference genome. And that's something you can easily do with this workflow here. So let's walk through it. So when you first click on the workflow, you have this handy overview page talking about some use cases, the summary of what the workflow is doing, all of the methods involved with references, as well as a handy flowchart diagram showing you each of the steps involved. It also has helpful input and output tabs, helping you organize your inputs and familiarizing you with outputs, but we're going to do all of that here. So let's just run through the workflow. Now, the first decision point you have to make is the type of analysis you want to conduct. Here, we're going to go with germline or diploid variant calling, but we have several options, including ancient DNA, somatic or human cancer variant calling, as well as even trio VCF calling. Here, germline will be totally fine. For the sequencing platform of your input data, Illumina, of course, is the most common and what we'll go with, but we also have options for PacBio or Oxford Nanopore long-read data. For the analysis mode, we're going to go with full analysis to show you everything, but we also can do this in piecemeal. So if you just want to check the quality of your input reads, we can quickly do that. If you only want to align your reads to a reference genome, we can only do that. And if you come with your reads already aligned to that genome and just call variants, you can also do that as well. But full analysis will do all three of these tasks. This parameter, if you come with lots and lots of data, it could take a long time to run. So if we have the option here to split all of your input reads into smaller files and perform the trimming and alignment in parallel, while this really speeds things up, it could be more costly on your compute time. And so for the purposes of this demo, we won't need it, but we do have it as an option for larger projects. So we need to now select our files that we want to use as input. Here under variant calling, I can just have my read1 and read2 files. So I will hit submit here. And most of the workflows on our platform require a design or an attributes file to orient to what the important features are of this data. So you can either create new or just select existing. Here, I've already made one for the demo, this design file, and you'll see what I mean here. So the first thing we need is the run ID. This is the prefix before your read1 and read2 on your read files. A sample ID, so what biological sample this is a part of. And your library ID, so what technical ID this sequence batch was run on. This is helpful for deduplicating reads at a later step, as well as a control ID, and this is really oriented to your question of interest. Here I just called it control in the group ID, but if I had two samples, it might be control and treatment, for example. So that's an example design file, and if you have any other questions on that, we have a lot of helpful documentation as to which analysis mode or input structure is best for you. So we can hit next, and we're going to select our reference genome as input. This is a human-based question we're working on, so we're going to keep the human genome, but we have a lot of different genomes here and can always add more depending on your question of interest. We can also limit the analysis by genomic regions. If you provide a bed file to zoom in on a specific chromosome or a gene of interest, in this case, we're just going to do the whole genome, so we don't need to do anything else here. Just hit next step. And this is where we can get a lot of our advanced configurations set. So the first is, by default, we select to trim adapters or poor quality reads. If you don't want to do this, these options will disappear for you, but it's highly recommended you do. You can select your trimming algorithm. Again, we have multiple options, but I'll go with trim galore. And we have some default settings here, just based on prior literature. Feel free to adjust these, but it's recommended to just keep with the defaults. Next, you can select your alignment output. We default to BAM as it's most versatile, but you could also output to CRAM if you need it for other downstream analyses. As well as selecting, next step is the alignment algorithm. So we have multiple different options for this as well. I'm going to select the Sention accelerated BWMM, but there's open source as well as other options. Next, from the alignment step, you want to have the option to mark your duplicates if you so choose. You can skip this step if you like, but it's often helpful if you would like to remove them at a later step. So here you have the option to only include unique reads in your QC and downstream applications, but for germline it's usually not an issue, so we'll keep that unchecked. And lastly, we want to select our variant calling algorithms, of which we have multiple options. I'm going to just select the non-accelerated deep variant, but you can select as many of these as you'd like, and the workflow will summarize them into a final union output, which I'll make sure to show you at the end. Well, we'll just go with one for now, a deep variant. Those are short read callers, short variant callers, and you can also do your longer structural variant callers. In this case, I'll select DELI2 for this example, but again, many options and lots of documentation on the side. So we'll hit next step here, and we'll be able to review everything that we've selected and make changes as we'd like. And now we'll give this a unique name. Here I'll call it variant calling 2 because variant calling 1 is already done, but I'll show you the output of. So here we'll hit run workflow. This ran for about an hour for me, and then we got this handy output. So this is what you'll see with a completed workflow. We have many handy HTML files summarizing the results. Here, one for the variants, showing distributions of variants by type. So we have SNPs and insertions here, as well as the classifications. So most of our SNPs were classified as missense variants, the genes that were most affected, allele frequencies, and TI over TV ratios. As well as an overview of your variants, we have an overview of your read quality. So here we have a FASTQ report. You would get one for each of the libraries you submitted, which would be ultimately summarized in this multi-QC report to give you a nice overview of the data. Here we have about 21 million reads, all of them mapping, and this is broken out by duplicates, multi-mapping, all of that. You get some nice coverage plots here, as well as insert size, GC distribution, things like that. But now if you really want the files to actually do other downstream processes with, they would exist in this All Files tab in the output here. Here's our HTMLs again, as well as the alignment BAMs for each of your input libraries, as well as total sample. They'll be aggregated together. Your input FASTQs for easy reference, and your final variant calls. Here's that union file I mentioned. If you had multiple callers together, they would all be concatenated here. But if you'd like the intermediate files from each caller individually, they're found here as well. So here we have our control deep variant. If you had a different group ID, it would be treatment deep variant, for example. And there's the original, the normalized, all these different intermediate files. But really the ones that you're most interested in are in those final variant tabs. We also have our QC data here and our structural variant outputs. So that's a brief overview of the genomics variant calling analysis, how to get from sequence reads to variable genomic positions with a click of a button. So thank you.